The true sign of intelligence is not knowledge, but imagination. This quote by Albert Einstein reminds me of our guest of honor speaker, Patrick Wong, because of his creativity of thought about research questions, design, and research challenges. I have tremendously enjoyed our 20 years of collaboration. Patrick holds the Stanley Ho Chair in Cognitive Neuroscience. He is professor of linguistics and professor of otolaryngology. He is also founding director of the Brain and Mind Institute at the Chinese University of Hong Kong, where he also serves as associate vice president of research. Patrick began his faculty career at Northwestern University, where he was a tenured professor in communication sciences and disorders. As a cognitive neuroscientist, linguist, and speech-language pathologist, Patrick's research covers a wide range of basic and translational issues concerning the neural basis and disorders of language and music. For over 20 years, his research team has been funded by NIH. His awards include NIH's Independent Scientist Award and Hong Kong's Research Grants Council Humanities and Social Sciences Prestigious Fellowship. In 2021, he was named a Guggenheim Fellow. So, Patrick, come on up.
Good afternoon, everyone. It is a real honor and privilege to be able to share with you some of our research that has been going on in Hong Kong and the US, some of that with Nancy as well. So I thank Nancy for the invitation and also the Society's invitation as well. Can we go to my slides? Oh, I press advance? Okay. Oh, can I go back? Well, anyway, so these are my financial disclosures. Oops. These are my financial disclosures. I'm a founder of a startup company in Hong Kong, initially supported by very generous startup support funding by the Hong Kong government. And also these are the patents related to the work that I'll be talking about very soon. So the work that we are conducting in my laboratory has this one goal. We're interested in identifying evidence-based solutions to optimize language learning for every child. So in the context of this goal, I would like to make 3 observations about child language acquisition. So the first is that in healthy children, language developmental outcome is variable, so that some of these otherwise healthy children would fall into a delay or atypical range. So let me give you 3 examples here. So each line here represents the learning trajectory of an individual child. And you can see that there are children here who are not learning very well, and there are children here who are learning very well. So the trajectory of development is quite variable, and you can see that across different language learning domains. So here I'm showing you the learning of vocabulary, but also tense marking, some grammatical markers as well. The second observation is that language development in children outside of the typical range is also variable. And also, if treatment is provided, their response to intervention is also variable. So let me show you some examples. I'm going to come back to this. This is about children with cochlear implants. So this is from Naparco studies from a while ago. So again, each individual line here shows the language learning trajectory of an individual child. And the yellow lines are the language learning developmental trajectories of children with cochlear implant. Here, what I want to emphasize is the variability that you can see. Again, you can see some children who are learning not very well, but also some children who are learning very well. I'm going to come back to this. And this is data from my lab. This is an autism intervention study that is currently under review. So as I said before, response to intervention can also be variable. So this is a clinical trial of autism that we conducted in Hong Kong. Again, each line represents the trajectory of an individual child. Now, this time going down is better because it means less symptomatic. And you can see that it's all over the place. Now, if we just look at the data in terms of like a traditional randomized clinical trial, we can see that this trial shows evidence of efficacious. So the treatment is efficacious. So this is typically what we want to see. But if you look very closely to the individual data points, you see that only about 60% of the children improve. Response to intervention is also variable. If we just look at the group, sometimes it's actually quite deceiving. Now, the third observation is this. Language development is stable in both typical and atypical populations under a business as usual development condition. That means if you don't provide special intervention, language development is stable. What do I mean by stability? So here, I want to show you just an example of 5 children, child A, B, C, D, and E. This is their language score. So at age 2, this is the ranking. So child E is the best in the group, child A is the lowest in the group. 5 years later, at age 7, these 5 children, they all improved compared to themselves, okay? But when we say stability, we're talking about relative ranking. So child A tends to be remaining in the bottom, and child E tends to be at the top of the class. So this is what we mean by stability. Not that the child doesn't improve, it's the ranking remains relatively stable. Now, this is group level results. It doesn't mean that there's no child A ever who will become child E. But just when you look at children as a group, if they start off low, they tend to remain low. That's what we mean by stability. We see stability across domains of development. We see stability across different populations. We also see stability in how parents interact with their children. So parents who talk a lot when the child is young continue to talk a lot to their children when the child is older. So we see developmental stability in the parents as well. And this is some data looking at preterm infants. So you can see that this is Mark Bornstein's data when he was at the NIH. So 5-month language data can predict 20-month language data, which can predict 4 years, 6 years, and 8 years. In other studies, he demonstrated that 6-month data could predict all the way up to 15 years. So this is evidence of stability. So what do these 3 observations mean in the context of our goal to find evidence-based solution to optimize language learning for every child? So developmental stability means that early language development can predict later language in both typically developing children and atypical populations. That is, if child-level prediction can be achieved. So today I'll be talking about how we make child-level predictions. And second, we don't necessarily need to wait for a diagnosis to begin intervention for those who may be predicted to be weak in language. That is, we assume that we can predict child level language success and also assume that we can find preemptive intervention that does no harm and that is cost realistic. Because if we prediction is not perfect. And so we'll be over identifying. So if we do preemptive intervention, that intervention needs to be cheap and it needs to not hurt the child. Now, I'm not going to be talking about response to intervention, but the same concept applies. So we can potentially predict response to intervention, and we don't necessarily need to wait for the intervention to complete to decide whether or not we want that intervention to continue. Okay, so this is another talk, but the same concept can apply as well. Now, why do we care about language? We know that 17% of children between 3 to 17 years have a neurodevelopmental condition. That's US data by CDC. So that includes primary language impairment, autism, ADHD, a number of different neurodevelopmental conditions. An important feature from my standpoint about these neurodevelopmental conditions is that language is one of the features. So in other words, if you can predict language, that could help you identify neurodevelopmental conditions early on. Now, for example, autism. We know that the number one reason why parents would take their child, autistic child, to see a pediatrician is because there's language delay. So knowing language is important to understand development. So in the rest of my presentation, I'll talk about how we predict, and then I'll also talk about what if we can predict. Why do we want to predict? Why is it useful? So I'll be presenting you with data looking at using neurodata to predict language delay in healthy infants and toddlers. Then I'll be talking about using neurodata, MRI data, to predict language development in atypical populations, including children with CI and preterm children. And then at the end, I'll talk about early intervention, like what I talked about low-cost intervention earlier. So what could be a low-cost early intervention? So let's talk about predicting language delay in healthy infants and toddlers. So I'll be talking about using neurodata. Now, the idea of using neurodata to make prediction is not new at all. So this is a theory developed by Patricia Kuhl at the University of Washington. So Kuhl has this theory called the native language neurocommitment hypothesis. So in this framework, what she's predicting is that you can use cortical data to predict language development. What kind of cortical data? So you can use something called MNN. So essentially, you can present children with their native speech and their non-native speech sound. And what she found was that a sensitivity to native speech sounds can be used to predict language development, vocabulary development more specifically. So this is one example in one of their studies. So you can see that children who has a really large gap here telling us that the child is very sensitive to the native speech sounds, these are the children who eventually develop language very well. Whereas children who pay more attention to the non-native speech sounds, those are the children who do not develop their native language as well, do not develop a vocabulary as well. So good detection of non-native sounds is a language learning liability according to our theory. Now, this is all very interesting, but this is looking at cortical responses, and so it's really hard to achieve child-level prediction. What I'll be talking about today is a method in which we can make predictions at the individual child level. So as you all know, the auditory nervous system is not just the auditory cortex. So from the ear to the auditory cortex, there are a number of different relay centers, including those you can find in the auditory brainstem and also the thalamus. What we're interested in is to try to find responses along these pathways, which can be used to help us make prediction. You're a doctor, so I don't need to tell you about the click ABR or the chirp ABR, the universal newborn hearing screening in many of the places in the world. You can tell me more about that. But what I want you to know is that this method is essentially using EEG because you use electrodes to try to see whether or not the child is sensitive to sounds, whether or not the child can hear. So this is the newborn hearing screening click ABR. What I'll be telling you about is a method that we use called speech ABR. Instead of a click response, we use a speech signal to help us understand the function of the auditory system of the child. The click ABR is a great screening tool because it can give you child level screening data. It can tell you whether or not Johnny can hear, the individual child, whether or not the individual child can hear, not a group of children. So for clinical purposes, it's very important to find interpretable data at the individual child level. So we're trying to do that, the same thing with speech ABR data. So what we do in this method is that we will present the child with some auditory signal, not click. We want to present the child with a rich signal that's rich in temporal and spectral cues. So for example, we can have the child listen to some music, but in our paradigm, we have the child listen to some speech sound. And then what we're measuring is what we call neuroencoding, how the child's nervous system encodes the incoming signal, speech or music that we play to the child. So let me give you an example. So here, the child is listening to something, and I'll be presenting to you results of two children and how the child's brain waves may sound like. Okay, so child A, baby A. So again, this is not what we play to the child, but how the child's nervous system encoded the sound. So baby A. Can you hear that? What was that? Oh, good. Okay. Because sometimes, like some audience, they can't hear. Okay, so good. So you can hear. So we play Twinkle Twinkle to this baby, and baby A encoded that twinkle twinkle, and then I play you back that waveform that we collected. Okay. Now let's listen to baby B. Okay. What can you hear? The twinkle twinkle too. Okay. So is baby A a better encoder or baby B a better encoder? Baby A. Okay, good. Okay. So, so this is neuroencoding. We get a waveform. The waveform is very noisy. Okay, so we've done a lot of processing and shrinking and expanding before we get to this point. But the point here is that there's individual differences in how babies encode the incoming signal, the incoming auditory signal. And from that signal, we actually get a lot of information about the function of that child's auditory nervous system. Okay, so if we give the child a speech sound with a varying pitch pattern, this is what we'll see. Now, these are average data. So for babies, this is babies encoding the Mandarin tone 3, a dipping tone. So it goes like ma, like that. So the black line is that pitch pattern. And the brown line is how children, this group of infants, encoded that signal. So you can see that these babies in general, follow this black line very well. Now, comparing to adults, adults are almost perfect on top of each other as a group. Again, we want to look at individual data, which I'll show you. But you can see that as a group, adults do better than babies. And the good thing about this method, there are many advantages of using this method. One is that it's basically the ABR that we are all familiar with. We just replace the click with a speech. And also, we can interpret the data at the individual child level and the individual listener level. Now, in our paradigm, we present children and sometimes adults with 3 speech sounds. The 3 speech sounds differ in pitch. So I'll be showing you data of Cantonese and Mandarin learning babies. In Cantonese and Mandarin, I'm a native speaker of Cantonese. So in tone languages, those are called tone languages, we use pitch to mark word meaning. So for example, in Cantonese, the syllable fu, fu, if you speak it with level tone, high tone, well, level tone 1, it means to hatch. Tone 2, it means tiger. Tone 3, it means pants. So fu, 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 okay. Fu, fu, fu. We have 6 distinctions in Cantonese. So I can say si, 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 si. Those are 6 different words. In Mandarin, ma, 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 ma. Those will be 4 different words. Okay, Mandarin is my other language. So we are seeing, so I work in Hong Kong, so our babies are all tone language learning babies, learning Cantonese in the next study that I'll be showing you. So what we wanted them to do is to listen to these pitch patterns, and we wanted to see if they're good encoders of these pitch patterns. As you can imagine, if you can differentiate pitch, you're going to be a good vocabulary learner. If you cannot distinguish pitch in Chinese, then you're going to be confusing tiger and pants. Okay. So in this study, oh, I can use the mouse. Okay, good. So in this study, oh, this is better. So in this study, we tested 423 children between 3 weeks and 24 months of age, and we gave them 3 pitch stimuli, three Cantonese syllable differing in pitch. And then it's a longitudinal study. We're following them for up to 5 years now. We have data for 3 years, including using the CDI, the MacArthur CDI, the Cantonese version, which is a parent survey of vocabulary, and also the Bailey. Many of you know clinically, Bailey you can use to assess development in infants and toddlers. So our question is whether or not we can use speech ABR collected from infancy and toddlerhood to predict language delay. So delay here is defined by 1 standard deviation below the mean. Language delay in preschool years at the individual child level. That's the question. And that paper has just been published in Pediatrics. So if you're interested, you can scan this QR code and go to that paper. So we use EEG collected between 3 weeks and 24 months to predict language in preschool years. Okay, so they listened to 3 GAs, 2 in their native language, Cantonese, and 1 in their non-native language. We presented them with a non-native tone in Mandarin, which is not in the child's language learning environment. And then also we collected information like sex assigned at birth, gestational age, birth weight, socioeconomic status, and other measures as well. And then again use them to predict the Chinese CDI, Bailey, and other outcome measures as well. Now we use machine learning, various machine learning techniques. I'll be showing you results of one. The important thing about these models is that we need to have external validation. Okay, so I mean, there are many ways of doing it. The essential thing about external validation is that the model that you are using, the data set that you use to train the models is different than the data set that you use to test the model. So external validation means you use another data set to test the model that you already developed, and you don't change the parameters of that, what we call internal model, okay? So I'll be showing you all the results that I'll be showing you will be external validated results. Okay, so let's look at here. So we constructed our predictive models using just what we call pediatric data. So we gestational age, birth weight, sex at birth, and just the EEG data, and then everything combined. So EEG data, pediatric data, and socioeconomic status. We had 423 subjects. 341 were born in public hospitals in Hong Kong. So Hong Kong has socialized medicine, so most children are born in public hospitals. Some wealthier families will decide to give birth in private hospitals. So we have 82 children born in private hospitals. So what we did was, one way of validation was to construct a model using data of children born in public hospitals, and then use private hospital-born children to validate the model that we constructed using children in public hospitals. So this is what the results here is. So if you look at, this is accuracy, sensitivity, and specificity, pediatric data only, EEG data only, and everything combined. So what you can see, I think sensitivity is important. So for screening purposes, so you can see that EEG alone can achieve almost 90% sensitivity. And if you put everything together, is not statistically better than just using EEG data alone. Perhaps because EEG already contained all the information about gestational age and birth, sex at birth, and birth weight and whatnot. So EEG alone can give us a sensitivity of 0.89. Now, again, that's 423 children. We're very happy about the results. And again, this is just published in Pediatrics. Now, these children, the youngest, were 3 weeks old. Okay, so they're not newborn. So what we are hoping to do now is to use the same method in newborn children. We want to see if data that we collect from shortly after birth can also be used to make predictions. I don't have the results yet. I'm going to be showing you some preliminary results. So we've tested over, so by the way, actually this study, we've tested over 1,400 children in Hong Kong and the Chinese mainland. And the data that we just published contain data of 423 children, but we have more data. And we've tested 500 neonates in Hong Kong and the Chinese mainland. And here I'm only showing you 54 kids. So these are their waveforms average across 27 subjects. Okay, this is another group of 27 subjects. And then we've been following them, and now we have some one-year data. And this is the group that shows evidence of delay based on the Bailey. They're less than 16 percentile. And these are children who are typically developing. Their Bailey is larger than 16 percentile, is better than 16 percentile. And so we're already seeing at neonates, their waveforms are different, their EEG waveform. So the SNR is way better in this group, in the group that's typically developing, than the group who are showing delay. Now, another way to look at these data is to use a method called Zebra. Okay, so Zebra is a neural embedding techniques to help us to look at neural representations. So remember, I told you that we presented these children with 3 different speech sounds. Okay, there are many trials that were presented to these children. So with Zebra, what we can do is to represent graphically how these children encoded these individual trials. So there are 3 colors because we asked them to listen to 3 represented to these kids 3 different types of speech stimuli. So there are 3 different colors: magenta, yellow, and gray. Magenta will be tone 2, yellow will be tone 3, gray will be tone 4. Those were the 3 stimuli that were presented to the children. And so if you see a ring, an oval ring like this is good because that means that there are good separation of those 3 different speech categories. So this is a child that has good separation. So the magenta is here, the yellow is here, and then the gray is here. So that means this child, this neonate more specifically, could differentiate those 3 different tones. And so the 27 neonates who ended up being typically developing in age 1 had rings like this, okay? So meaning, again, good separations of the 3 tones. Whereas if you look at the 27 who were language delay, you can see that their representation is in a circle. That means there's a lot of overlap among the different categories. So it seems like we're still following these children, but it seems like even in neonates, we could see differentiation, good encoding, good differentiation, and that could become, hopefully, that could mean that those are markers of whether or not they will develop good language later on. We only have 1-year data, but we keep following them. And so hopefully in a couple of years, the 500 children will all be followed. So my interim conclusion is this. So in seemingly healthy infants, we can predict language delay using an EEG test that is similar to one being used in standard newborn hearing screening. Our predictive models survive external validation. I don't have time to talk about the various hypotheses, but we've developed a new hypothesis about the division of labor of the auditory brainstem and the cortex in terms of language development. It seems like it's supporting our newer hypothesis about the division of labor of subcortical and cortical centers that underlie language development. And what we're doing now is to develop a prediction model for newborns in public hospitals in Hong Kong and Shenzhen. Shenzhen is in the Chinese mainland for both Cantonese and Putonghua learning infants, neonates. Now, we are also using the same method to look at autism as well. And so this is a paper that we published a few years ago, and it seems like the same method could help us differentiate typically developing children and autistic children who are learning Cantonese. And we're following up on that. So that's our study looking at, I have 5 more minutes, or 7 more minutes. So that is our study looking at infants and toddlers using EEG. So now I want to tell you about our studies looking at children with cochlear implants. So as you know, auditory and spoken language abilities and aspects of cognition are better in children with cochlear implants than children, their peers, who have hearing loss without cochlear implants. But compared to normal hearing children, children with CIs, in terms of these aspects of cognition of language and language, are still lagging behind. Now, these processes are not observed by ears, they're observed by the brain. So what we're interested in doing is to look into the brain to try to use brain data to construct a predictive model of language development in children with cochlear implants. This is a study that we've been doing for the past 15 years with Nancy and other colleagues as well. So I want to go back to this graph that I showed you earlier. So this is, again, from Naparco and colleagues. So each line here represents the language learning trajectory of an individual child. And so the yellow lines, again, are children with cochlear implants, and the gray lines are normal hearing children. If you look at the average, the group average, you see what I just said earlier, that children with cochlear implants, their language learning is not better than their normal hearing peers. Now, to us, the more interesting thing is the variability. Okay, so here you can see that the variability is quite large for children with cochlear implants. Children with normal hearing, they are variable, but not nearly as variable as children with CI. So in our research, we are finding ways to help us predict this variability. We don't want to predict variability when variability happens. Okay, we don't want to predict variability at this point. We hope to predict variability at this point before we can observe variability. So in our study, we use 3D brain scans collected before surgery to help us predict spoken language outcome after cochlear implants. So clinically, many centers, not all centers, would collect brain data. And now we're going around the world literally to convince people that, if you put the child in the MRI scanner anyway and collecting some brain data, you may as well collect three-dimensional data, which can be used to build these predictive models that I'm about to tell you. And as you know, the kids will come back for various assessment at various time points, so we can use those data points to help us construct the predictive models as well. Now, this is a study that we published a few years ago, comparing brain anatomy of children who have normal hearing and CI candidates. The CI candidates all have hearing loss. That's before they get a cochlear implant. These are anatomical scans. Now, when you compare the two groups, you can already see anatomical differences. This is in terms of density. So, for example, the auditory cortex is thinner in children who have hearing loss than normal hearing children. And so in that study that we published in, that was 2018, we constructed a very preliminary model to try to understand whether or not these areas are most predictive of language development in children with cochlear implants. That is, areas that are affected by hearing loss. Or they're actually using areas outside of the auditory cortex, areas that are not affected by hearing loss, to help them develop language. So that's a while ago, and now we have a lot more data. Now, in our ongoing studies, we're gathering data from 5 US cochlear implant centers, the University of Melbourne and the Chinese University of Hong Kong, where I'm at, to construct bigger predictive models. We're interested in whether or not we can use brain scans, anatomical scans before surgery, to predict whether or not a child is a good improver or poor improver of spoken language. So a binary classification. Now, there are a lot of challenges. One major challenge is that if you look across practice around the world, they're slightly different. They may use a different scanner, the assessment protocol may be different, the surgeon is different. So there are actually many different things that they may differ. And so the challenge here is, how can we build a single model that's useful for all clinical practices rather than just having to develop a model at each individual center? So the results that I'm about to present to you were just published in JAMA Otolaryngology a few months ago. So if you're interested in the full results, you can go to this paper. So there's a lot going on here. I'm going to walk you through the slide in a minute. So we have different data sets, as I told you earlier. So here we have a set of data from Chicago, from Nancy's practice. These are all English learners. We also have English learners from Melbourne. We have over 100 subjects provided by them. And then also we have Spanish learning children also in Chicago, in Nancy's center. Now, we could use the same machine learning techniques that I told you earlier in the EEG study to construct these models. So if you look at the performance of these models, all external validation, so you can see that single data sets, we could construct pretty good models. So sensitivity is always over 80% in classifying whether or not the child is a good language improver or poor language improver. Accuracy is good, specificity is good, AUC is all good. Okay, now single data set. That is, we use Chicago data to test the Chicago English model. We use the Melbourne data to test the Melbourne model. Okay, even though it's a data set that withhold, but still it's the same center. Now, the real question is whether or not that one model is useful if we use data that's far away from that center. So for example, here we use the Melbourne data to test the Chicago model, okay? Or we use the Chicago Spanish data to test the Chicago English model, okay? And then the most challenging case will be to use the Hong Kong data, different language, different center, to validate the Chicago English model. So you get what we're trying to achieve. Now, again, single data set, beautiful results. Look at the cross-center, cross-language data, okay? It's chance performance, crap, okay? Not good, okay? So it's telling us that maybe we need individual center models. Well, but this is the good news. Now here, when you look at the final box, if we combine our data sets all together, if we put the Chicago and Melbourne together, we put the Chicago, Melbourne, and Hong Kong together, the models are doing pretty well. So sensitivity, again, is going back to over 80%. So the lesson here is that if we all work together and try to contribute data to build heterogeneous model, then we could potentially have one ginormous model that works for everyone. Now, we are working very hard. We're literally going around the world to try to get more data. But at the same time, we're also developing better machine learning methods as well. So here in my lab, Simon Wong developed this directed evolution model method to try to improve transfer, improve generalization. And so far, remember I told you that we were at chance level when we are trying to cross-validate. But in the new method, we can improve our model's generalization by 15 to 25%. So if we have better method, better data set, we could potentially achieve a really good prediction across centers. Now, we are actively looking for new collaborators, so please join us if you're interested. We need 3D brain volume, brain MRI before surgery, and then good follow-up after surgery. So please let us know if you're interested in joining our study. This is the QR code that you can scan. Okay, I have no time left, so I do? Oh, I do? Oh, good, okay. Okay, good. Oh, okay, yeah, I'm gonna slowly move to the other slide. Oh, okay, so I thought I had 30 minutes. Okay, good. Okay, so we're using the same kind of conceptual framework to look at preterm infants. We're especially interested in moderate to late preterm infants because that's the group that's very variable. So some of them develop language and cognition well, some of them are not doing so well, and you can't tell until they are 5 or 6. My children were born preterm, and then things start to surface when they have to learn how to read. Okay, so, and then when they have to sit still and try to pay attention in class. So this is the range that we're interested in. We're interested in using EEG data and also MRI data to make prediction as well. So this is a study that we're conducting in Hong Kong. In the US, you have really big infant imaging groups like Minnesota and UNC. You already have really good tools. In China, when we try to do this study, we have to start from scratch. And so the first job that we had was to develop brain templates that are suitable for analyzing brain data of Chinese babies. So that's what we did in the past few years. So we've scanned over 300 babies, but 224 of them were born preterm. And so from that data set, we constructed the first Chinese infant brain templates in Atlas. They fit our brains better than when we used the UNC template. The UNC template is a little bigger, the frontal lobe is different, and so with Chinese brain template, we can more accurately and automatically extract brain areas from Chinese babies' brains. And we're using this to construct predictive model. And what we found is that if we use white matter, we can achieve pretty good sensitivity in predicting whether a child is going to have language delay. So same kind of logic, but instead of applying it in children with cochlear implants, we're doing it on children who are born moderate to late preterm. Okay, so we can predict. So far I've been telling you about EEG and MRI and demographic data, constructing predictive models, externally validated predictive models to predict whether or not a child would likely have language delay up to age 3. We're following them for longer term, and then to see if prediction still holds. Now, the important question is this to parents. It's not prediction. So to parents, they want solutions. They don't want prediction. So prediction is useless unless there's a solution. So what is the solution? So let's talk about that. So from decades of research, child development research, language acquisition research, we know that high quality caregiver-child interaction can promote language development. For some reason, some parents just naturally know how to interact with their child in a way that will promote the child's language. So what we want to see is something like this, that you read to your child, you play with your child, you respond to your child, you expand the utterances of the child, you recast. There are techniques that we know that some parents would just do, and those techniques are associated with better language development. What we don't want to see is this, okay? So basically anesthesia. Here you put the child in front of the TV, then you're fine, you don't have to do anything. So these are the children who don't develop language as well, because the TV is taking away quality care-child interaction time. So that's there are papers in recent years trying to explain the mechanism behind why watching TV is not good for the child at that kind of age. It's because it's taking away time from interaction. So now the good news is this. We know that caregivers can be taught. We can teach them techniques to promote language of their child. If they do that at home, you're going to get a really high dose of intervention. Okay, if you just send the child to a speech pathologist, it's going to be 1 hour a week. Okay, so that dose is not very high. If you do that at home, it's naturalistic, and then the dose is high. So the intervention that I'll be talking about is to teach parents how to do this at home. We want every parent, every caregiver, sometimes it's not a parent, sometimes it's a grandparent, there are diverse families. So we want to teach the caregiver to do this. Now, if caregivers learn how to do this is a meta-analysis by our colleagues, Megan Roberts. She showed that in the meta-analysis that if parents do this at home, you can teach parents how to do this. If we teach parents to do this effectively at home, on average, you can get an effect size that can be over 0.9, almost 1 standard deviation. Okay, so that's a really huge improvement in the child. That's receptive language. Some of you are speech therapists, you know that receptive language is very hard to treat, okay? So if you do this early in infants and toddlers, receptive language can be 0.9 on average. Now, we can teach parents how to do this. And now in Hong Kong, let me talk about this first. Oh, you said I have time. Okay. Oh, now okay, good. So I need to wrap up. So we have good evidence suggesting that this is effective for children with primary language impairment, children with autism, and children who are hearing impaired. In Hong Kong, we've tested a method in which we deliver this entirely online. We teach groups of parents how to play with their kids essentially online, and it's effective. Now, I'm wrapping up. So now, across China, what we're doing is to test a newer model in which many of you probably watch short videos on your phone. So we created 600 short videos to teach parents how to do this, okay? So, and then they watch, so this is a dad, we have dads, it's important to have dads in these roles, grandparents, and then that. So here the dad is playing with the child, is talking to the child when a child is giving a bath to the child. So you can, if you have WeChat, which is the most popular social media platform in China, you can go into our program. We're testing this towards universal implementation doing pilot testing across a number of different Chinese cities and rural areas. So my conclusion is this. So we are now in the position to use neurodata to make individual child prediction about the child's language development. And potentially we can use that prediction and pair that with low-cost intervention so that all children's language development can be optimized for that particular child. Thank you very much. So these are my former and current lab members involved in the study, my collaborators, and funding agency, including the NIH. Thank you.